So the pancreas, we learned about in the GI system, has an exocrine function of secreting digestive enzymes into the GI tract. But the pancreas also has an endocrine function, and so it's responsible for secreting a few hormones, specifically insulin, glucagon, and somatostatin. So this is a picture of the pancreas. We have the exocrine ducts, acinar cells. We have the ducts where the exocrine pancreas works. And then we have the endocrine pancreas, all these islet cells. These are called pancreatic islets, and we have cells in the islets. And then these cells make these hormones. So the alpha cells make glucagon, beta cells make insulin, delta cells make somatostatin. You don't really have to pay attention to this. Okay. So now let's talk about how insulin is made in the pancreas. So again, what cells makes insulin? Remember, beta cells make insulin. So first, from DNA you get RNA, and then you get that polypeptide. So this dark red stuff is the stuff that's going to become insulin. This thing here is going to be a C peptide. As you can see, it looks like if you fold it this way, it looks like a C. And then here we have a signal peptide. So this is an N-terminal signal peptide, and it's going to direct this pre-proinsulin to the rough ER. So this thing right here is called pre-proinsulin. Okay, it's directed to the rough ER. It's taken up by the rough ER, and then once it gets into the rough ER, the signal peptide is cleaved. So this thing is removed. As you can see here now, there is no signal peptide, and this is now called a proinsulin. Previously pre-proinsulin, now proinsulin. Okay, pre is like before it becomes proinsulin, and now it's finally become proinsulin. And through what? Through cleavage of the signal peptide. So step 1, step 2 is cleavage of the signal peptide. Step 3 is now your proinsulin is transported to the Golgi apparatus. This is this, and it's packed into secretory granules. So that's step 3, transport through the Golgi apparatus, package into secretory granules. And these are the secretory granules. And now you're going to have cleavage of the proinsulin inside the secretory granules. That's going to lead to formation of insulin here. Remember, it's the dark red stuff, and you're going to get C-peptide. So that is how insulin is made. I just want you to know that every time you make insulin, you get a C-peptide. So pre-proinsulin, step 1, directed to the rough ER by the signal peptide. Step 2, pre-proinsulin loses signal peptide, becoming proinsulin. Step 3, proinsulin directed through the Golgi apparatus into the secretory granules. Step 4, inside secretory granules, proinsulin is cleaved into insulin and C-peptide equally. Insulin from the pancreas goes into the bloodstream, goes throughout the body to exert its effects. And now we're going to talk about insulin effects now. So insulin binds to insulin receptors throughout the body, and the main function here is to lower blood sugar and promote growth. So lowers blood sugar and growth, means anabolic stuff. So it basically lowers blood sugar by causing cells to take up glucose and use that glucose to help build things. So it's promote growth, anabolic. It's going to build things like lipids, like proteins. It's going to make glycogen. Okay, and it's through these two pathways, the P3K and the RAS-MAP kinase pathways. So I want to add that these effects are tissue specific. So for example, I told you how about it's going to help make lipids, make proteins, but it doesn't make it on all cells. It acts on fat tissues for lipid synthesis. It acts on skeletal muscles for protein synthesis. It acts on the liver for glycogen synthesis. So these can be any cell. I just want to go through this picture. Insulin binds to its receptor, causes a lot of signals, causes the P3K pathway, the RAS-MAP pathway. P3K pathway causes vesicles that are carrying GLUT4. GLUT4 is a channel for glucose. It's going to cause translocation to the cell membrane. So now you get these GLUT4 channels in the cell membrane, so that glucose can be taken up by whatever cell that insulin acted on. This can be any cell throughout the body. Okay, so that's how you get glucose, and then you also get glycogen, lipid, and protein synthesis, whatever that cell needs. And in the RAS-MAP pathway, you have DNA synthesis and cell growth. Okay, so that's all to lower blood sugar and promote growth. Now, additional functions of insulin include cellular uptake of amino acids and potassium. And remember, you might have we might have learned this before. Remember that beta agonist and insulin make potassium go into the cell. And the way I remember the beta agonists work, it's like kind of I tie everything together. Remember insulin, what cells in the pancreas make it? Beta cells in the pancreas make it. So and the way I remember is beta cells as well as insulin make potassium go into cells. Okay, the other function of insulin is it decreases glucagon release from the pancreas. We're going to talk about that in a little second. Glucose is basically the opposite of insulin. Now I want to talk about insulin release. So we talked about how insulin functions, but what causes it to be released? The main regulator of insulin release from the pancreatic beta cells is glucose. So we talked about how insulin is made. We talked about what happens when insulin goes in the blood and acts on other cells. But it's first made in the pancreas cells, and it's hanging out until glucose goes ahead and triggers insulin release. Glucose goes to the pancreatic cells and triggers release of insulin so that it can go into the bloodstream. Now there's a few other regulators of insulin release, especially in the GI system. Do you remember what they were? Specifically, it was GLP and GIP. So GLP was glucagon-like peptide, GIP was glucose-dependent insulinotropic peptide. So these are released after meals to help with glucose intake. It makes sense. You ate food, you ate a bunch of carbohydrates, you had ice cream, it goes into your gut. You have all this carbohydrate there, and you want to take that into your body, so you're gonna stimulate release of insulin through these different molecules, GLP-1, GIP. And hence, will you have a greater insulin response to an oral glucose load or an IV glucose load? So let's say you make someone either swallow 100 grams of glucose or you directly input 100 grams of glucose into their bloodstream, which will lead to greater insulin. Remember that glucose is the main regulator, but the glucose that goes through the mouth goes into the GI tract and triggers release of this. So oral glucose has higher insulin response than an IV glucose. So how exactly does glucose lead to insulin release? What is the little cellular mechanics of all this? In the pancreatic beta cell, we have in pancreatic beta cells, we have insulin stored in these little vesicles. Glucose, we said, was the main regulator. So glucose goes into the pancreatic beta cell. It undergoes glycolysis, so you get extra ATP generation, you get energy generation. So now you have an increased ratio of ATP to ADP. Okay, this increased ratio will lead to inhibition of this potassium channel where potassium leaves. So now you have too much positive charge. You have depolarization of the membrane. Okay, depolarization of the membrane causes you have voltage gated calcium channel. So you just change your voltage. You have depolarization. Now this channel gets activated. Calcium can come in, and calcium is going to stimulate the release. It's going to stimulate the release of this insulin from the pancreatic beta cell, and now insulin goes into the bloodstream and then goes throughout the body and it exerts its effects. It goes into the muscle, goes to the fat, goes to the liver, and that is how insulin is released. So we have now covered how insulin is made, we have covered how insulin is released, and we have covered what will happen once insulin gets to different cells. Remember that insulin, what was the main function? It was to lower blood sugar and promote growth. So that makes lipids, makes proteins, makes glycogen. Finally, let's go to glucagon. We said that glucagon was the opposite of insulin, and it is made by the pancreatic alpha cells. So if it's the opposite function of insulin, what will glucagon do? What is it? How does it affect blood sugar? And how does it affect, does it cause catabolysis or is it anabolic or catabolic? So blood sugar, it increases blood sugar and it's catabolic. So catabolic means you're going to break down stuff, and the whole point is you break down fat, you break down muscle to increase your blood sugar. So overall, you get glycogenolysis, you get gluconeogenesis, you stimulate lipolysis, which helps with glucose production. And this is what you see. This is just a little picture of what I just told you. Glycogenolysis, gluconeogenesis, and you get lipolysis. Fatty acids help you break down fatty acids into acetyl-CoA. Acetyl-CoA is going to be turned into glucose. And the other thing you're going to see is well, the acetyl-CoA can also make keto acids. So when you get glucagon, you increase glucose in the blood, you increase fatty acids. That's from the lipolysis. And you increase keto acids. Okay? Increase glucose, you increase fatty acids, you increase keto acids. Now, what is the regulation of glucagon? Will low blood sugar or high blood sugar cause release of glucagon? When will you want to increase blood sugar? When you have low blood sugar, let's erase this so you can see this. When you have low blood sugar, then you want to increase your blood sugar, and you do that through glucagon. Now, inhibition is through insulin. We talked about that. Somatostatin pretty much inhibits everything. And then obviously hyperglycemia then would inhibit it. So that is glucagon. When glucagon increases blood sugar, is catabolic. Insulin decreases blood sugar, is anabolic. Alright.